Welcome to the Tech Talks Daily Podcast, where you can learn and be inspired by real-world examples of how technology is transforming businesses and reshaping industries in a language everyone can understand. Here is your host, Neil C. Hughes.
Welcome back to the Tech Talks Daily Podcast, and a big hello to the team at Lemon Pie for their message that they sent over saying here's to changing the world with one podcast episode at a time. And as regular listeners will know, I'm fascinated by our new proactive rather than reactive approach to healthcare and the role that technology is playing in that change. For example, with just a watch and a smartphone, we can now monitor our steps, exercise, heart rate, track calories, and even have an ECG machine on hand 24/7. But when I heard about a company called Glympse, which is on a mission to detect diseases earlier and help develop better therapies so that people don't become patients and patients are returned to health faster, I had to find out more. And through their technology, activity-based biosensors are bioengineered and tunable to any protease-mediated disease. And Glympse's technology can measure and predict the trajectory of a disease in a non-invasive, real-time functional readout that is clinically actionable. And how does that work? Well, this is what we're going to learn about today. So buckle up and hold on tight so I can beam your ears all the way to Washington, D.C., so we can speak with Caroline from Glympse about all this and much more. So a massive warm welcome to the show, Caroline. Can you tell the listeners a little about who you are and what you do?
Great. And thank you very much for the invitation to be here today. Delighted to be here. So I'm Caroline Loew. I'm the CEO of Glympse, which is a Series B diagnostic startup that's based in Cambridge, Massachusetts in the US. So my background is that I have over 20 plus year career in drug development in the pharmaceutical industry. And that has actually, spanned a number of different areas. It's included experience in regulatory and market access and commercial. And most recently, I was at Bristol Myers Squibb, where I had the number 2 position, the R&D organization. And what really kind of drives me is kind of solving our problems, actually, just this whole notion of what, science can do to, transform sort of diseases for patients, really. And, sort of this, fundamental belief that you can bring, really smart people together and they can focus on hard problems and we can make a difference for patients. So that's sort of what makes me get out of bed. And I'm very excited to be doing what we're focusing on at Glympse.
And it's that fundamental belief that is one of the reasons I invited you on the podcast today. And I am conscious we will have people listening and hearing about Glympse for the very first time today. So for those people, can you set the scene and tell me a little bit more about the kind of problems that you're solving with your technology, and also a little bit more about Glympse's overall mission to, like you say there, detect diseases earlier and ultimately help develop better therapies so that people can become patients and patients are returned to health faster?
Mm-hmm. So, disease can be really hard to detect and also to monitor. And, I think we've probably all experienced this in some form or other, right? We probably all know someone, like a family member, a mom or a dad, a brother or sister who's been on one of these terrible diagnostic odysseys, right? They start to feel unwell, they get sick, they go on this terrible journey where they're trying to find out what the problem is. And kind of at the heart of that is, and it takes years sometimes, right? And we all know it and we all hear about it. And when they finally get, diagnosed, the disease is quite advanced. Sort of at the heart of this is that there just aren't very good tools to diagnose disease. I mean, surprisingly, right? We have many great medicines. We've made many great advances in medicine, but we don't actually have the best tools to diagnose disease. And that problem is not just then about, finding out what's wrong with patients, but it actually impacts drug discovery and development. That alone is a really complicated problem. And we know that it's littered with failures. And those failures are in part because we can't identify the right patients for drugs, and we can't tell whether, early on in the drug discovery process, whether we have what's called target engagement, whether the drugs that we identify are actually impacting the right biological target. And so there is kind of this whole set of problems that are associated with not having good diagnostics. And so our technology is aimed at solving that, and aimed at solving that in a very broad, sort of biologically fundamental way. And our technology is protease-mediated. We have in our body, naturally over 570 proteases, and they're present in every aspect of human health and disease. They're involved in sort of every part of how our bodies operate. And so what we're doing with our platform is we're taking advantage of that. We're taking advantage of something that is present naturally, biologically, and we're using the pattern of those proteases that are present in a specific disease state, and we're monitoring how it changes over time, dynamically, real time, to measure disease activity. So we can tell sort of baseline what your disease state is, and then how that activity changes, sort of how severe your disease is, and then if you're being treated, whether your disease is progressing or regressing. And so we're using what's naturally present in your body, we're figuring out how to measure it, and we're sort of tuning it to a specific disease state. So that's sort of in essence what our technology is, but it's aimed at solving something that is really kind of a big gap in how patients get diagnosed and treated today.
And that's one of the things that really fascinates me, because I think traditionally we used to have a reactive approach to healthcare where we waited for something to go wrong and then try and find a solution to it and diagnose the issue, whereas now we're much more proactive. And one of the things that stands out about what you're doing at Glympse is these activity-based biosensors that are bioengineered and tunable to any protease-mediated disease. But can you tell me a little bit more about the technology that makes all that possible?
Yeah, absolutely. So sort of at a really high level, what we can do is with these biosensors, right, these we call them biomarker cocktails, right, we can detect real-time biological changes in a disease using these sensors. And the sensors are bioengineered, and they're tunable to any protease-mediated disease. And basically, most diseases have some protease-mediated component to them. And we can then non-invasively query, this disease activity. And the great thing is that we can tune these sensors to each disease state. So what we do is we go through a discovery process. For a specific disease, we spend time understanding what proteases are present in the disease state. We then build a specific kind of a custom panel, if you want, of sensors that will query that disease state. And then how this works is once we've built that custom panel, we inject the sensors into the body. They go to the disease tissue. They measure the activity of the proteases that are present in the disease. And then within an hour, the patient, we just collect a urine sample from the patient. And we do a standard lab analysis, what's called a mass spec analysis. There are many different types of analysis we can use. We use a mass spec currently. And then we just analyze using a proprietary classifier. That classifier is built to the disease state. And then what gets reported, and this is what the physician sees, this is what the patient hears about. Most importantly, a clinically actionable result. So there's actually information that comes out that says, your disease state is, severe or moderate or whatever. It's something that's very actionable, and the patient can actually do something with. And this is really, really important. Real time output in terms of the disease state.
It's incredibly cool. And as you were talking about it there, I was getting flashbacks of that Inner Space movie 30 years ago. I'm probably revealing too much about my age there. But do you have any use cases that you could share that just bring what we're talking about to life?
Yeah, there's a disease called NASH, which is a fatty liver disease. And it's actually been called kind of the silent killer. It's a disease that in many patients kind of emerges over the course of like 10 to 20 years. So it's a slow evolution. In the US alone, there are around 20 million patients with this illness, and currently there are actually no therapeutics for it. There are lots of companies, 60 or 70 companies developing therapeutics for it. But in its more severe forms, patients actually become cirrhotic and their livers fail, and they have to have liver transplant, or they die, unfortunately. So it's very it's a very significant liver disease. So the problem with this disease is that there really aren't any good diagnostics for it, and so finding out whether you have it is really problematic. And if you can detect it early, you can actually. And in its early stages, it's believed to be reversible, and there are probably things that you can do about it. Once it becomes committed, you're just on a trajectory eventually to, towards liver failure. So the question is, how do you diagnose it? Today, hard to detect. The standard of care is actually a needle biopsy, which is truly as horrible as it sounds. You inject a small aspirated needle into the liver to take a tiny piece of the liver, and it's looked at under a microscope. And this is it's a painful procedure. It can't be repeated. And once patients have it, they generally don't want to have it again, and many won't even consent to have it. It's a really uncomfortable procedure, and it's not very accurate, as it turns out. So we're actually developing our first indication is actually for liver, for NASH, for this diagnosis of this liver disease. And we've been able to do two very cool things with our diagnostic. The first is that we can, we appear to be able, with quite high discrimination, tell between all of the stages of this disease. So it's a progressive disease. It goes from very mild to very severe. And we can tell all of these stages quite clearly in our early data to date. So that's very positive. And the second thing is that we can also tell dynamically what's happening. And this is very important. So what do I mean by that? I mean that if your disease is getting worse, we can actually see that very progression very clearly. And if your disease is getting better, we can also see that progression. So for a patient who is undergoing treatment eventually, and we're working with drug companies who are developing treatments, we can help them understand very quickly, is the treatment working for a patient? Does the patient need to go on to another treatment? Which is very important. Now, I mean, you could probably relate to this, and I think most people can. You want feedback if you're undergoing a treatment. You want to know, is it working? Is it not working? Should I be switching to something else? And so this becomes a very, very valuable tool. So that's our first indication. And that gives you a sense of how a diagnostic like this, a simple non-invasive diagnostic, can replace, a very invasive, candidly kind of impractical diagnostic that is currently available to assess a disease.
It's incredibly cool, and it seems like there's just so much room for opportunity here, and I'm curious where it could all lead to. So can you expand on the pipeline applicability of your technology and what other diseases you can study with it? Because like I said, it just seems there's so much scope here, isn't there?
There is a lot of scope, and, we have to be focused. And many diseases are protease-mediated, as I said. So we have a lot of opportunity, and we have proof of concept in over 10 different diseases. So a lot of choice. But we're focusing next in cancer and infectious disease. And, in cancer, we're focused on a very interesting opportunity. So there are lots of, very specific cancer diagnostics. They're genetic in origin, often very specific to the cancer. We focused in a slightly different area, which is sort of around this idea that our diagnostic is dynamic, so that we can detect, very, very clearly kind of real-time changes in the tumor status. And when we combine that with a treatment, we're able to see, again, from early data, we're able to see very early whether a tumor is responding to treatment. And so if I can give you a kind of a practical example of this, you may have heard of immunotherapy. It's been kind of the major advance in cancer treatment in the past few years. And typically what happens in immunotherapy is that patients are treated, and somewhere in the region of sort of 9 to 12 weeks, they'll go back and they'll have a scan, and they'll be able to see whether they're responding to treatment. Now, 9 to 12 weeks in cancer is a lifetime, particularly when patients who are being treated with immunotherapy are often have very severe, fast progressing cancers. What we've been able to see in our early data, and again, this is something that we're still developing, is that we can see response to treatment within 1 to 2 weeks. Now, this is the kind of thing that if it ends up being clinically applicable is game changing, because we could tell very early on whether a patient is responding to immunotherapy, and instead of them continuing on that course of treatment for 9 to 12 weeks, having a scan, seeing if they're responding, if this turned out to be valid, or this type of application turned out to be valid, if they weren't responding, which is the most important situation, they could actually switch to another therapy. And you're buying them, 8, 10 weeks on a second line therapy or third line therapy, which is so important when you're treating a significant disease like cancer that where every day is important.
And I'm curious, how safe are biosensors proving to be in these human trials? Is there anything you can share on that?
Yes, so in clinical development, the drugs go through 3 stages, or 2 to 3 stages of development generally. But the first stage is always a safety study, where you test the drug in a very controlled manner in healthy volunteers to make sure that it's safe. And we completed those studies last year, and we presented the data at a major scientific meeting. And our drug was extremely safe, had very, very high safety margins, and that reproduced the data that we had also seen in our preclinical studies. And so the great thing with this is that not only is it kind of like essentially non-invasively going and doing kind of essentially like a blood test on your disease state inside you, right? It's measuring real time kind of the activity of your disease inside you, but it's also extremely safe as it's doing that. And it's essentially, the way to think about it is it's non-invasively doing that. So everything that we had hoped for came out in those studies.
And something else that I think is incredibly exciting with what you're doing here is I believe you're using machine learning techniques to refine future readouts of biomarkers too. Can you expand on that?
We are. There are really sort of 2 aspects to this. In our core technology today, we aggregate data. We build we use machine learning, relatively straightforward machine learning, to build a classifier that we use to analyze the biomarker data that we capture from each test. And that gets refined over time. That's a relatively straightforward machine learning technique. Perhaps what's more interesting is that over time, as we capture more and more of the data, we're actually building on a unique area of biology. We're working in a new biological domain of protease biology, and we'll be capturing that data with structured clinical data. So what you want to think about is patients' blood pressure and basic data on their blood chemistry and so forth. And so we'll have other structured clinical data. And when you have aggregated structured clinical data like that, together with unique areas of biology, and it's sort of anonymized data sets, you can do deep machine learning. And you can then accomplish kind of 2 things. The first is that you can much more fundamentally understand disease states, and then you can start to make predictions about patients based on their diagnostic results. So an example might be the kind of thing that I think we would expect to be able to do once we have large volumes of data is we could look at a patient's diagnostic results, and based on the body of data that we have previously analyzed, we could probably say, a patient based on their diagnostic test would be better suited to use one therapeutic over another based on the body of data that we've seen previously. And so this kind of data is very powerful, and we're certainly very excited for the potential that it will offer in terms of improving patient outcomes.
And there are so many things to be excited about here, especially about the future potential. But what is it that excites you about the road ahead?
You're right. The platform has a lot of potential, and I think obviously the most exciting step for us right now is the process that we're in of gathering clinical data on our NASH indication. In any drug or diagnostic company, we're sort of a combination of both. That's always the most exciting step that you go through. But I'm I'm candidly looking beyond that to some of the other indications that we have under development that are following quickly. And I'm very excited about that. And that's the nice thing about having a platform. You're not sort of you're not just focused in one area. And the potential to have this broader impact is very exciting.
Now, a question I always like to ask my guests is if there was a particular song that inspired them in the past, helped them on their career, helped them get their head in the zone before going on stage, or just reminds them of technology. So my question to you is, what would your song be and why? And before you answer, I've got to say, I'm going to be disappointed if you don't say Cupid by Sam Cooke, just because of the inner space reference. But is there anything that springs to mind?
Oh, my. It's a funny question. I think people, I certainly use music, and I think a lot of people do. People use music for a lot of different things, like to relax, to energize you. And for me, I find that there are phrases in songs that I really love that mean a lot to me. Yeah. And there are lots of songs that are part of special times. And I would say for now, one of the things that helps me a lot in my work, in fact, for a long time, I've meditated. It's part of how I work and how I focus. But it's not always possible. They're just like the pace of life. But there's certain music that I actually find really helpful in achieving kind of a calm and a focus. And one of the songs that I always use to kind of like get me in that mode is a song by Chicane, No Ordinary Morning. And I listen to that and I can just I can switch and I can get into kind of this very focus zone. And so whenever I need to focus, whenever I'm trying to get in the zone, that's the song that I listen to.
I love that. A great choice. And I will be adding that to the Tech Talks Daily Spotify playlist too. But before I let you go, though, for anyone listening that would like to find out more information, contact your team, or especially if they've got any question, what's the best starting place for Glympse?
Sure. So we're on Twitter and LinkedIn. So at Glympse Bio on Twitter and on LinkedIn, and I'm at Caroline Loew on Twitter and LinkedIn as well.
Well, I always say at the end of every episode that technology works best when it brings people together. But what you're doing here by detecting diseases earlier and helping develop better therapies so that people don't become patients and patients are returned to health faster with technology at the heart of all that is incredible. So a big thank you for sharing that with me today.
Thank you. It's been a lot of fun. Really appreciate the time.
I love how Glimpse's technology can detect changes in the site of the disease without having to do surgical procedure. And also intriguing how they're building this data engine that will ultimately use machine learning techniques to refine future readouts of biomarkers. All of that is incredibly cool. And like I referenced in the interview, bits of it did remind me of the 80s movie Inner Space. But over to you. I want you to share with me your thoughts on today's conversations. Send over any questions you've got or even song suggestions for the Tech Talks Daily Spotify playlist. You can do all that by emailing me, techblogwriter@outlook.com. Remember, my website is techblogwriter.co.uk, where we'll have over 1,550 interviews of this podcast, plus the show that I host with Citrix called Tech Fusion. And finally, just a genuine thank you for me for tuning to listen to this podcast every day. I'll return again tomorrow with another guest, but until next time, don't be a stranger.
Thank you for listening to the Tech Talks Daily Podcast with Neil C. Hughes. Remember, technology works best when it brings people together.